All right, everybody, welcome. Happy December 1st. Today's webinar is with Elucidata, and the topic is utilizing public transcriptomics databases for meta-analysis. We're going to have three speakers and guests, and we will introduce them shortly. So today's agenda, we'll give a brief industry perspective. We'll do the speaker introductions, and then Elucidata will give their talk and conversations, and then we'll go into a Q&A. If for any reason you have to leave early, this will be recorded. You could add your questions to the chat or to the Q&A, and they'll get answered at the end in the Q&A session. It's pretty straightforward, and looking forward to hearing what Elucidata has to say. So, brief industry perspective from 2015 Visioneers. What we wanted to do is kind of level set for everybody who may be on the call that maybe doesn't have transcriptomic experience, etc. So do some definitions, give you some background. So the transcriptome is a set of all RNA transcripts, including coding and non-coding, in an individual or a population of cells. This field, to be honest, has exploded over probably the past 5-7 years. So generating lots of data, lots of insights. The whole multi-omics space is going to start to answer and provide insight, answer questions and provide insight into biology that for many years people just said, hey, we just don't understand the biology details. We just don't know. I think we're going to start to see in the next several years a lot of great discoveries and insights from a lot of this basic research. It is the study of transcriptomes and their functions. The field of transcriptomics allows for the examination of whole transcriptome changes across a variety of biological conditions. So if you look at the image, you know, it's basically your RNA, you start to produce proteins from that RNA, and then you start to affect the metabolome through sugars, nucleotides, amino acids, lipidomics, lipids, et cetera, et cetera. And, you know, this is going to, in the next 5 years, there's going to be more boxes added to this space as discoveries are made. So what can it tell us? An RNA sequence mirrors the sequence of the DNA from which it is transcribed. Okay, so basic biology. But by analyzing the entire collection of RNA sequences in a cell, the transcriptome, researchers can determine when and where each gene is turned on and off in the cells and tissues of an organism, which starts to get at the root of disease states, etc. So this is very critical, and this is, again, providing insights at a molecular level that scientists and researchers just didn't understand several years ago. So there are several techniques in today's laboratories: microarrays, which quantify a set of predetermined sequences; RNA sequencing, RNA-seq, which uses high throughput sequencing to capture all sequences. And some key methods when it comes to mRNA sequencing, to RNA sequencing. So you have mRNA sequencing, you have targeted RNA sequencing, ultra low input and single cell RNA-seq, RNA exome capture sequencing, total RNA sequencing, small RNA sequencing, and I'm sure I've missed something. But you also have express sequence tags, you have SAGE, which is a hybridization-based microarray, you have real-time PCR, and you have RNA interference. Now, what comes with all of that is lots and lots of data, and you really want to be producing model quality data with provenance, with that's fair, findable, accessible, interoperable, and very importantly, if you do all that, reusable. Data, scientific data, is just another form of currency, and if we mismanage that, we have problems, and we're going to have problems. But with this new multi-omics approach to things, we're going to have serious problems after investing lots and lots of money to produce all this data. So that's when bioinformatics tools come into play, and especially for transcriptomes analysis, etc. So there are a lot of great tools out in the market today, and you're going to get to hear next about Elucidata and what they're doing to manage this and perform awesome analysis. So Maya Stedman is a bioinformatician at Elucidata, working with data scientists at therapeutic and diagnostic companies to identify and resolve roadblocks in their research due to unclean biomedical data. It's a huge problem in our industry. Prior to Elucidata, Maya was a metabolomics scientist studying cancer and autoimmune disease at several biotech startups in Cambridge, including Agios Pharmaceuticals. And while at Agios, she had the chance to work with Elucidata's founder, Abhishek, who taught her that the value of your results are determined by the quality of your data. Wise man. And Yogesh is a product manager at Elucidata with a focus to solve problems associated with data findability and data management. He works closely with the search, storage, and library teams within Elucidata. And before that, he has spent around 4 years building point-of-care medical devices to strengthen public health infrastructure in India. He studied at MTech and BTech in biotechnology and biochemical engineering from IIT. Nobal is a solutions manager at Elucidata with years of experience in creating engineering workflows and data products in bioinformatics. At Elucidata, Nobal is involved in delivering bioinformatics solutions to his customers and therapeutic and diagnostic companies in the form of custom automated workflows, analysis pipelines, and visualization dashboards. And with that, I want to turn it over to this great team.
Thanks so much, John.
Yep.
Oops, wrong button. I always do that. All right. Should be good here? Okay.
Good.
As John said, today we're going to talk to you about utilizing public transcriptomic databases for meta-analysis. And as we get into it, I want to spend a little bit of time talking to you about what meta-analysis is. So when used properly, meta-analysis can lead to key insights in the drug discovery process. And what it actually is, people think meta-analysis, they understand kind of conceptually, you're talking about looking at multiple studies at once, but true meta-analysis is a systematic and formal way to do that. And when it's done properly, you can greatly improve your statistical power and make really in-depth insights that would normally take multiple iterations of an experiment to do. And that kind of leads us into why meta-analysis is so important. As John alluded to, there's so much data out there, but individual studies tend to be pretty limited in sample size, right? You can't often get the statistical power that you need to come to really concrete conclusions. And so one of the ways that you can improve your statistical power is by combining multiple transcriptomic studies together. And in addition to not just improving the accuracy and reliability of your results, you're also able to make more generalized results about that body of research. And so now that you kind of understand like the basics of what meta-analysis brings to the table, when we talk about the outcomes, most commonly we're talking about differentially expressed genes and the pathways with which those genes are in. And so differentially expressed genes it's a basic comparison, A over B, 2 cohorts, but getting there is a lot more challenging than it seems on its face. And in addition to the genes that can go on to become biomarkers and drug targets, coming back to those pathways, the pathways that the genes are in can give you real deep mechanistic understanding. So not just I have a list of targets that I could potentially go after, but true hypothesis generation and understanding of the system that you're studying, right? So, anyone can come up with a list of the top changing targets, but to have the contextual understanding behind why those genes are important or why that pathway came up and what that has to do with what you're studying is where meta-analysis really shines. And again, this all sounds great, well and good, but it's not as easy as it sounds. And I think that you guys know that, or you wouldn't be in this webinar with us today. And the problem is that the biological data landscape is very fragmented, right? There's silos of data all over the place. And even when you do find excellent repositories for data that you're looking for, such as GEO, and there's a number of others that we at Elucidata use quite frequently. I'm going to pick on GEO a lot today, though, so I'll bring it up in the beginning. The data that you find there, it's in different formats, there's different syntaxes, and those create logistical and technical challenges that require deep understanding of not just the data, but also bioinformatics expertise to overcome. So all of that is just to say is that although multi-analysis studies are common, truly integrating them and doing them properly is really requires a substantial amount of expertise. So throughout today's presentation, we've decided to frame it in terms of coming back to a biological question that you may ask in your day-to-day research, and then applying how you would first attack this problem generally and outside of poly. And then we're going to walk you through sort of the improvements that we've made to this workflow here at Elucidata and the insights that it's led to. So our biological question today is very basic, right? What are the top differentially expressed genes in melanoma? That's our disease of interest. And there we go. And so it all starts, like I said, with selecting the right data for your meta-analysis. Here on this slide, I've got the basic steps to performing analysis, meta-analysis. We've started with our biological question. Next, we're going to identify our relevant pool of data. And that can even get broken down further, right? Any one of these steps, there's countless individual parts of it. So identifying that pool of data is you start with your potential pool. You have some quality criteria. Again, this still looks pretty straightforward, right? Just my disease type, the data type, the types of samples that I need in that study to get to your high quality data that you're then going to take forward, clean and enrich, perform your analysis to derive insights. And I'm going to focus a lot more on this findability problem because this is where people often don't get past. And if you can't get past finding the right data, your meta-analysis is going to be for naught because you're not going to have the correct information to go forward with. And so what it comes down to is public data is available, but it's not fair. And John had defined it earlier in his introduction, but I think it's worth stating again that fair is findable, accessible, interoperable, and reusable. And if data isn't fair, the relevant data sets that you need to come to your correct biological conclusions, not only are they not going to be identified, they're just not going to be used at all. And so we've started with our high-level search, again, with melanoma, that was needed to have that disease in there. I need control samples because I know I want to do differential expressions, so I need that baseline. And let's say I also want a drug treatment in there because it's reflective on the type of analysis that I want to do later, or maybe I want to add some more in-depth analysis. And then finally, it's something that I need, you know, about the types of measurements that are involved, right? So making sure that the metadata is actually complete, that the genes that I'm interested in, that maybe I have preconceived hypotheses are there. And those form our exclusion criteria. And so as we go from the top high-level search that's pretty broad, and we start filtering that data out, there's a huge rate of attrition. And in addition to just losing data sets based on them not meeting those high-level criteria, you also start to lose data sets through that manual review process. Perhaps you read the abstract and you realize it's not quite relevant. Maybe they mention melanoma or they study melanoma, but it's the hypothesis doesn't align with what you're trying to study. And in addition to true exclusions, because the data is not relevant, you also are excluding data based on lack of information. And that incomplete metadata is, frankly, it's unacceptable because we should be in a day and age where you can trust the search results that you get. And so you're kind of in this tug of war between trying to find the data that you want effectively and programmatically or using informatics tools, and then needing to manually review all of those data. So if you start super broad, you're going to have to read, you know, thousands of papers, and that's not feasible. And if you start narrowing down, you have to understand that you're going to be excluding potentially relevant results based on a lack of standardization. And so what do we mean by standardization? This slide here, I know is very wordy, but the key takeaways is that when metadata aren't standardized, without human understanding, domain expertise, and that context piece, you can't use it, you can't take it forward. And so in that first box, this upper left-hand box here, we have hepatocellular carcinoma here. I know I said our example was melanoma, but forgive us for this one, we're using a slide. The mesh term for hepatocellular carcinoma is that top list. But me as a researcher, I know that any one of these diseases could be relevant for my meta-analysis. Programmatically, I can't possibly include all of these into my search, or I'm going to have an unruly amount of data that I have to filter. But then also manually, I can't possibly look through each one of those papers because you're just going to spend all of your analysis time trying to gather the data before you even get to analysis. And that's just one part of the problem, and I won't dwell on this too much more, but understand that in addition just to terminology differences for finding valid data sets, you also have like super annoying things like extra spaces or a hyphen or a spelling error. And all of this lack of standardization in the public sphere is what makes data unusable and is one of the first challenges that people often don't even get past when you're trying to do a multi-analysis, a multi-study analysis. And so this is where I get excited because Elucidata, here at Elucidata, we're experts in data curation, and we have firsthand felt this problem. It's why the company was founded. And so I get to ask Yogesh the question now, is there a better way to find public data and to do this more efficiently?
Yeah, so thanks so much, Maya, for setting the context of how hard this problem actually is. And with this, I would just like to share my screen and take you through some of the slides. Yeah. Before we go into the demo, I would briefly talk about the technology which we are building at Elucidata. I will break it into 3 steps. First, curating the data from public domain at scale. And here the scale is really important. And getting the curations right is equally important. So what we have done is we have built those extract, transform, load ETL pipelines, which allows us to get the data from the source. In this case, we'll talk a lot about geo today. And then the next step is actually performing standardization on that data and then performing like harmonizing the metadata, following ontologies the way we curate it so that the downstream consumption part of it is really streamlined. After we find the data and get it from the source using the ETL pipelines, the next step is to store the data onto the data infrastructure, right? And that is very, very important for you to be able to access control as well as reuse the data in future, right? On Poly, later in the demo, I'll show you how you can perform a bunch of very powerful queries once the data is on the warehouse. And finally, from the warehouse, you can plug in multiple tools, be it a GUI application or a code or any bioinformatic pipelines or machine learning models to perform the consumption downstream. Now, let's talk a bit more about how the standardization and harmonization part is being done. So at source, you will find a lot of metadata just missing, or you'll have to actually spend a lot of manual effort to be able to curate it. And none of it is harmonized. Everyone talks about a disease or different context in different words, and there is not a real standard way of defining and not at all something which a machine can understand. And that's the problem of curation, which we are solving by putting appropriate structure and harmonizing the data. So you can see that on Poly, the bunch of metadata fields which we curate, like disease, organism, tissue, drug, cell type, cell line, data type, and source, and many more, like all of these are properly harmonized following standard ontologies, machine readable, and we provide outputs in consistent GCD files. And having a structured file format really helps you scale up your analysis. Yeah, so without any further ado, I would directly hop on to give you a small demo of the problem statement we started off today. right? So in today's problem, we are trying to find differentially expressed genes in melanoma. So more specifically, we want to find clinical data where the problem statement is find disease samples versus normal samples, and then create those cohorts out, and then perform meta-analysis on top of it. Now, this looks pretty straightforward. There's a search bar on top. You can use multiple operators to drive your search. On the left panel, you have a bunch of filters. These are curated fields. And I'll start off with writing the search keyword, right? For me, I'm looking at clinical data, be it patients, be it donors, like there are different ways in which different people write. So I was just writing a bunch of them in OR query. And then we are doing the AND, that we want something healthy, control, and also performing melanoma on top of it, right? So once we query this, once we search this, this is all powered by Elasticsearch on the backend. Once this is done, I'm going to use, so you can see there are 299 data sets, which is the first output, but this is not specific enough. So what I'm going to do next is try to find diseases which are related to melanoma. Now, in here, as you can see, the results are classified into two categories. One is matched results, and second ones are related. Now, the difference is that all these are direct matches of the term, whereas the related category is something which is not melanoma exactly, but a neighbor as per the mesh ontology. So we apply these filters out, and the next filter I want to go to is tissue. Given melanoma is related to skin, I'm just going to search for skin tissue, and in here there are a lot more related tissues. So I'm going to just pick all of them and then apply these filters. And finally, I'm just going to pick organism as Homo sapiens. Now, after doing this, what I have is 37 data sets, and I can quickly read, and these are all from Geo, so I can quickly read the summary, the design, and be able to perform analysis on top of it. One cool thing we also have on the platform is being able to open applications to perform analysis. This is a bit out of scope of the demo, so I'm just going to not go over it. But now this was the GUI first way of being able to find data. I'll get to more exciting, more challenging, which is being able to try to get to it in a code first manner, right? Please feel free to drop your questions on the chat box, and we'll be happy to take it up at the end of the session. So as you know, so far, a bunch of metadata fields we curate at a data set level, and each data set has multiple samples for which we also curate a bunch of sample level metadata. Right. So before I go further, I would like to take a few seconds and talk about the Omics Atlas warehouse, which we have, data warehouse, which we have. In that warehouse, there are three tables which is of prime importance. One data set, sample, and feature. And this is a block diagram of how these tables are related to each other. For a given GSE ID from Geo, that information is stored at the dataset level, and then for each for a dataset there will be a bunch of samples. Then for each dataset the samples are stored in a separate table, and then corresponding to it there will be features which are stored separately, and then there is data matrix. So data matrix is all the normalized counts for across different genes being studied and across different samples, right? So this is sort of the framework of how the whole Omics Atlas is set up. So I'll take you through at a high level what the schema looks like for all these tables, right? For Geo specifically, so we have fields such as abstract, author, a bunch of curated fields as you saw before. We also capture a few fields from source, which is really important for findability of data, such as description, experiment design, summary, and like link of publication and so on. So it becomes really easy to navigate through all of this. The next is the schema of the sample level metadata, and like people who have worked with Geo, you would know that like there is a lot of unstructured material out there. And so in sample level, we have not indexed the entire sample level across all the data sets, just identified few which we have indexed, which you can query upon. And like we have also curated few fields on top of it so that findability gets easy. But at a high level, we have some labeling protocols, treatment protocol, extraction protocol, growth protocol, etc., which are commonly published by the authors. Now, let's look at the query. So in here, we are trying to identify like these metadata fields, data sets, curated disease, curated tissue, description, from geo dataset table, where the disease is like matches melanoma. So you remember on the GUI when I was showing you match category and related category, the same thing you can perform on the SQL query as well. So here the disease is matching to melanoma, and then tissue could be skin related tissues, right? And then organism Homo sapiens, transcriptomics. And this is a query where we are looking at like these keyword list of our interest, right? And in these keywords, we are trying to search it across description, summary, design, and abstract from the publication. And we are performing an OR operation here so that any of these along with this will come out in the output. So once we query this, we get around 174 data sets, but this is not specific enough. Remember the problem we started off with? We wanted to compare healthy versus disease, right? Now, just looking at the data set level metadata won't get us there, because out of these 174 data sets, there will only be a handful of them where actually a disease and normal is being compared head to head. Now, how do we do that on Poly? So this is where a sample level metadata comes into picture. So as you know, like at sample level, we store the info, curate the disease for each sample in the data set, right? So as you can see here, we are looking at a case where if the disease is normal, then we label it normal samples. Otherwise, the disease, if other diseases, then it's disease sample. And we are performing a query within a query, right? So you can see that in here, we had around 174 outputs. These were all melanoma specific. So we are just pasting the previous query here, like, and because you can join the sample table with dataset level table, it's fairly straightforward to do. Now, as you can see here, like these are the data sets which are the result. You can see for each data set, like how many disease samples, how many normal samples, and your cohorts are really very clearly defined for you to take it through the analysis, right? So you have 21 data sets, which we have identified. And for these data set, I have just like put this data frame out here where you have description, design, summary, number of samples. And I understand that, the notebook is not like best suited to read these text heavy fields, so you can export it in a CSV file. You can and then you can dig deeper from there. Now, this was like identifying the data sets, right? So let's go a little further deep into some of the sample level queries, very high level. Like I'm looking at all the samples in the geo sample table where the disease, where the curated disease is melanoma. And there are around 11,000 such samples, and it was so easy to just fetch all of them. And then you can see around 10K of them are of melanoma. Then there are other matches of melanoma which also are there. And these are spanned across around 600 different unique dataset IDs. And just last part of what I want to communicate is being able to query the data matrix, right? So once the experiment is performed, you have normalized the counts, you have the data matrix. And in the data matrix, you basically have different samples in the columns and different genes in the rows, and you have the expression values in the actual matrix, right? So in here, you can just see that these are the list of genes of my interest, and for one particular dataset ID, I'm just trying to query the expression value of all these different genes. And you can see that like it took me around a second and a half to fetch the expression values of these genes. So I think it's very powerful for people to identify data sets of relevance. Yeah, so I'll pause here, and after we have found the data sets of relevance, I would pass it over to Noble to tell us about how exactly we performed the meta-analysis after the data findability problem was solved. So Noble, over to you.
Yeah, thanks,
Actually,
Yogesh.
Can I actually really quick interject just for 1 second? I noticed that there was a
Sure.
question in the chat that was specifically about our curation and how we're coming up with those with the tags. And so the question specifically was about the ambiguity around a tag, right, when we're doing our curation. And I thought that was closely related to the findability question, right? Because the quality of the tag itself is about our curation is gonna determine how accurate the search is. And so just wanted to quickly answer that we use AI, right? So we use NLP models that are doing the curation. And then there's also a layer of quality assurance. So we have expert curators that are going to review those suggested tags. And we use 2 independent curators to review those results. If they're in agreement, it's accepted and moved on. If there's a disagreement, there's a 3rd independent curator that comes in and reviews that. And these are also active learning models. So as the more data that those models see and the more corrections that are made by our expert curators, the better that they've become. I'm also going to plug that we also have a white paper coming out very soon specifically about the quality of our NLP models. So you can look forward to that. But this is a great question. And there's a lot of information that we have on our website about our data quality and our quality assurances when it comes to curation. So I just wanted to interject that there. Anyway, on to you, Noble.
Thanks, man. Thanks, Yogesh. Yeah, so Yogesh talked about how we find the data sets on Poly. I'm going to talk about the rest of the workflow where we will run differential expression analysis, and then we are going to store the results in another atlas, which will allow a user to make queries against genes or against a particular perturbation versus control comparison. Right. So this is like a very high level overview of what the workflow looks like. So essentially to start with, we have this RNA-seq datasets available on Poly, which has been curated. And then we are essentially going to look for specifically those datasets which have a very simple design, where we are simply doing a perturbation versus control kind of experiment. Because there are a lot of the studies which also have multifactorial design. We are not going to include those ones. For example, some of them can be about time series experiments as well. So we are not going to go into those complications for the sake of this demo, right? And then we are specifically going to look and find out data sets where melanoma was compared to its normal counterpart. And then there's another block here where we're essentially running the differential expression for melanoma versus disease across all the data sets that we have on Poly. And then the data output of the differential, essentially the result of differential expression, is stored in another atlas. Why we are doing that? Because the user will be able to query, for a particular data set or for a particular comparison or a gene. After that, we further go one step ahead, where we also perform another downstream analysis, which is doing the gene set enrichment analysis on top of the existing differential results that we have already. Now, we are also going to store the results of those GSEA computations, which essentially is about, your enrichment scores, the p-values, the log FC values, etc. Right, so again, I will switch back to the notebook here. All right. So as we talked about that, the data in Poly is stored in a data warehouse, which we are calling Poly. And within Poly, we have multiple atlases which are storing the data. Whatever differential results that we are generating by running the ETL pipeline, the end output is actually stored in this atlas called GDX atlas, which has 2 tables. One is the data sets table and other one is the features table. I will talk about what those two tables are and what kind of data do they store. So data sets table is essentially the table about the differential comparison itself. So any given data set on Poly can have multiple possible comparisons. Now each row in this table is essentially one differential comparison, and it also has additional attributes for the comparison, like what was the perturbation, what was the control. And remember, the perturbation and control that we are using here are properly ontologically curated. That, which is why it will make it possible for a user to simply enter ontological valid mesh term and find out all the comparisons corresponding to that perturbation versus control experiment. Now, another table that we have here is the features table, which is essentially storing the results of the differential computation, such as the name of the gene, the associated log2 fold change values, the average expression, p-values, district p-values, etc. Another key thing to notice here is that apart from this results, we are also storing the source dataset ID, which is essentially acting as a foreign key, so that we can perform a cross join on the datasets table, which is having the metadata about the differential comparison itself. So these are the two tables that we are going to be using for performing all our queries on the data. Right. Now, as Yogesh talked about, that we are essentially going to perform an analysis where we are where we intend to find out what are the different biomarkers or the genes which are highly expressed in melanoma versus normal comparison. Now, doing this kind of analysis is possible on this atlas because all the data sets have been properly curated, and we are only restricting the perturbation control names to be following that standard nomenclature as defined by the ontology terms that we are using for disease. So we essentially start off by looking at what are the different comparisons that we have available in our data warehouse, which are about melanoma versus normal. Turns out we have about 20 such data comparisons, differential comparisons, which are about melanoma versus normal. Now I will look further into those comparisons and extract out what are the genes which are differentially expressed in both directions for this particular comparison of interest. Now, this is one simple query where we are essentially extracting out the log fold change values, the p-values, and the other associated matrix with the differential comparisons for all the comparisons for this melanoma versus normal. Right. Now, here I am taking a very simple approach where I am simply looking at the log2 fold change mean values on an average. So first, I am filtering out only the genes which follow, which have a p-value of less than a threshold that I have specified here. For example, I'm going to look at only the genes. I'm filtering the query results only for the genes which have a p-value of less than 5%. And within those, I am now ranking the genes by their average log2 fold change values. Now, once I do that, then this is the result that we get for this comparison. Turns out TNC, PRX1, EMP, PRAME are some of, in fact, these are some of the well-known biomarkers that are available out there for melanoma versus normal comparisons. Yeah, and after this, we have another step in the journey, which is after we have found out the data, the genes, we are also interested in looking at what are the different biological pathways which are enriched in this particular comparison. So just like this notebook, I have another one which is pretty much the same, except that looking at the results of differential computation, we are going to look at the results of the GSEA analysis here. So most of this notebook is going to be the same. I'll just scroll to the bottom of this page where we are going to look at what is the different pathways which are enriched for this particular comparison. And one important thing that I forgot to mention is that like for every gene differential comparison that we have in our GDX database, we are actually running GSA analysis for more than 20 databases available, in public domain. For example, we are running it on the Hallmark GeneSet database. We are also running it on the KEGG, Reactome, Wikipathways, Gene Ontologies, etc. So as an example, this is the results that came out from the GSA computation which was run on Hallmark GeneSet database. So as we see that TNFA signaling via nuclear factor kappa B is one of the highly enriched pathways that is there in melanoma versus the normal skin comparison. Right. And similarly, we can also look at the results that we have from some of the other database. For example, here I have plotted it for the KEC database. So like the KEC chemokine signaling pathway and the KEC RNA degradation are some of the important pathways which are highly enriched in melanoma versus the normal comparison. Now, another thing that I wanted to show is that I will switch back to my previous notebook here. Now, since the data is available in Poly in a curated format, it is very easy to extract results for this kind of experiments. So as an example, until now I have showed you like how to get the list of genes given disease. I can run actually a live example here as well. So I will simply need to go and change the disease of interest. For example, instead of melanoma, I want to look at what are the genes which are highly expressed in, say, neuroblastoma. Since the data is curated on Poly, I can simply replace the term here, and let's see what we find out for this particular comparison. Yeah, so we see that FOX2B, FOX2A, HIST1H1B are some of the prognostic biomarkers that we find to be associated with neuroblastoma. Now, what I have shown here in these two demos is barely scratching the surface. Now, you can use the same workflow to do other kind of searches as well. For example, if you are interested in finding out what are the genes which are differentially expressed in a drug versus untreated comparison in a specific disease context, that is also possible to do, only because all the samples that we have on Poly are curated, be it for drug, be it for disease, cell line, cell type, etc. Now, let's say you have a hypothesis that regarding drug candidature, for say melanoma. So what I will do is I will take the list of genes that you found in melanoma as highly differentially expressed and see what are the different drugs that we get as targets here. So what I can do, I can switch back to another notebook here, where I am essentially taking the list of genes that I got in my first analysis. So these are some of the genes that we saw were differentially expressed in melanoma versus normal. Now, this search is different. Rather than finding out the list of genes given a disease versus normal comparison, we are actually trying to find out what are the drugs given a list of genes here. So these are the genes that were differentially expressed in melanoma, and most of this cell is essentially the same thing, querying the data warehouse to find out the different drug combinations. And I will take you to the bottom of the page here. Right. So these are the different drugs that were identified as like they are the drugs which are down-regulating the set of input genes that we have provided. So, like for example, we see some of the drugs which are already being used to treat melanoma. For example, imidacloprid and sorafenib, vorasidenib, etc. So these are some of the drugs which are already being used to treat melanoma patients. So yeah, this is something that you can do with the data that is available in Poly.
Noble, thanks so much. I had a quick question actually that I think was earlier on in your demo here. When you have the table of calculated results, I noticed that it was this one right here, right? So you've got your log2 fold change. I think there was one also that had p-value. Is there any limitations into the types of statistical methods or the analyses that you can do on those results? Are you limited to just doing a basic log2 fold change in this structure?
No. So since we are storing the results of the differential analysis as log2 fold change values, p-values, FDR, etc., we can pretty much use any kind of advanced or sophisticated meta-analysis techniques. For example, I can do a rank sum product or a p-value combination techniques to do that, only because the data is stored and available for the users to query from the atlas.
That's awesome. And then I think additionally, I thought maybe we could either save it for the end and have a more specific discussion on it, but I thought maybe you might want to comment on sort of concern that people might have about combining multiple analyses that maybe have different hypotheses or different experimental methods, right? So that's kind of an underlying, we didn't really touch on it in the beginning, but now that we're looking at these use cases, you know, how confident can I really be in these results, considering the fact that there's multiple sources that are leading into it that we're using, right? By combining multiple studies, what does that say about our confidence in the results? Should I take it less seriously, more seriously? I don't know. I thought, as you are the SME here on this one, I thought I'd
That's a very good question, Maya. Right. So yeah, I have seen a lot of people raising that concern that data is quite heterogeneous, in geo, and how like confident can one be while doing any of this meta-analysis. Now, I would rather argue that since the data that we have is quite heterogeneous, any signal that is real is going to stick out, and whatever we are getting, despite that existence of heterogeneity, is something real and not likely just by chance.
Yeah, I think that's something that like there, in my previous life is before I had access to this type of so my dog's itchy now. I hope you can't hear that. Before I had access to poly, my previous life as a metabolomic scientist, I often was very afraid of combining even more than one of my own analyses, right? That I generated, I was confident, same method, because I didn't feel confident in my ability to overcome sort of that experimental difference. But when you're really looking at the data like this and you see these results come out, it improves your confidence quite a bit. Okay, I think, were you all set with, is there anything else that you wanted to show here? Or should we jump to the takeaway slide? Great, okay, so I won't read this slide out all the way through, but I just wanted to take a couple moments and open it up to questions here and put these up to kind of prompt discussion. When we were making this presentation and going through these demos, these were the types of key takeaways that really stuck out to us, and we hope to you as well. And the top, the top point is that when it's constructed properly, a well done meta-analysis can not only lead to new hypothesis generation, also hypothesis validation and a deeper mechanistic understanding of the body of research that you're looking at. But that's really contingent on having high quality data that feeds into that analysis. And, we've covered that the struggles with identifying the data that's available publicly and how much work goes into handling that manually, and hope that you have seen, we've been able to show you and demonstrate that the effort that's put into curation really improves our ability to have rapid insight generation by storing this analysis ready data that you can iterate on, as Noble showed us in that use case there. And so Yogesh and Noble, if there's anything else that you want to add on here, otherwise happy to take more questions. I think that's all from us.
Yeah, we do have a couple more questions. I know there was a couple, Yogesh has answered a couple behind the scenes, but there was one left over from Andrew's questions before around when you're doing a lot of this cleaning, cleansing, fixing of data, etc. Do you ever have to reach out to researchers in their papers or their work and say, hey, we're trying to do this? I thought it was a really good question because it's not something that maybe people do. So I was curious what the answer to that was.
Right, it's a great question. And so, I think that everyone who has tried to get more information from an author kind of can encounter a similar thing. A common thing, not necessarily around the context, but maybe around missing metadata or a missing supplemental file. That's also pretty common. And we do, when we notice that those data aren't available, you can make the effort to reach out. Oftentimes you don't get a response. Luckily, there's a lot of data that is available that can be curated. And in addition to, when the author is not available, our domain experts, it's not just one person. These are highly trained people with lots of experience in, biomedical research that are able to derive context. And, we're really reading these papers. We're not just looking at the tables that are attached to these data sets and going from there. There's context applied from the author in what they've written. So I hope that adds a little bit more
Yeah, no,
confidence.
it's real. It's the real world that we live in. So great. And as everybody was talking, all I could think about is, target identification, validation all the way through. And I see that. I see both that capability within your platform, but also, when you're also at the later stage of drug development, et cetera. So do you guys want to talk about that a little bit on the power of poly and how it's used in the industry? I thought it was kind of pretty awesome how I could see it used across the continuum.
So I would like to take a stab at that. One of the things in my experience talking to a lot of industry folks is that they want to derive insights from multiple places and see what comes out very often. And often they want to, derive insight from public domain, validate through experiments whether it matches from what their proprietary data is. This is where poly comes into picture. Like you find data from public domain, analyze it, derive your insight. But at the same time, if you store that data on the Omics Atlas, in the way that whole infrastructure is designed, you can actually very easily perform analysis with both the public data as well as proprietary data combined. And that is when you get like real sense of um, what's the confidence on the data set, on the insight which you have generated when you have verification from multiple ends, and on poly it becomes really easy to do that.
Okay.
Noble, Maya, if you want to add to that.
I think one of the things that stuck out to me, if I would just add on, is how much data you can go through so quickly, right? Like, I think that's the biggest jump that we made. And I know you just kind of touched on there, but that was for me leaving, working with just my own data, just my proprietary data, and using papers just to validate the results that I was generating in the lab, but to flip that on its head and be able to go the other way and then take the vast amount of data that's available publicly and use my limited time and resources in the lab to validate those results so efficiently. I think it was great.
Yeah, no, it's pretty cool. All right, there's another question from Andrew. Are you accepting each data set's expression normalization or renormalizing to your favorite genes?
Right. Yeah. So we have our own EDL pipeline, which starts from the FASTQ files, and it processes all the data sets in a standard and consistent manner. So whatever data we end up storing in our atlas has been run through a very standard pipeline.
Okay. So I saw an interesting, the public data to curated and the amount of data that's remaining. I think it was about an 80% reduction. Did I read that properly, Maya? Was it from that graph?
Yeah, that's based on a couple of different studies that
Yeah.
we've done, but roughly
Well,
it's a pretty large attrition rate.
Well, the truth is, so based on all of our work that we've done with clients all around the world, that's true internally many times for large pharmas as well. So we get asked a lot, hey, could you do a data assessment for us to determine if we should migrate this data? What should we do with it? Is it have quality, et cetera, et cetera? And we normally find about 80% of the data is not of a level of a high enough quality or enough metadata, et cetera, to make heads or tails of it. And it usually goes by the wayside. And so it was interesting to see, even with the public data, the numbers are, it's a shame to see, I shouldn't say it's interesting, it's a shame to see that even internally at some companies that they're still at an 80% quality problem and it's not reusable. So this is a really interesting point for why fair data environments are so critical.
Yeah, and John, I would just say I was able to share it in the chat. We've got a lot of, I said it earlier, but there's a lot of documentation on our websites and a lot of, and there's another webinar that I gave a little while back about ensuring data quality and how to improve that ROI, right? To go from this attrition and the true effect, the monumental effect that standardization, harmonization, and engineering can take on improving the retention of data and improving quality. And it's all based on having that context, right? By putting your metadata in with the biological context and applying the data and the metadata together, you get a lot more use of your data set, and you're able to hold on to it for longer.
Yep. I don't see any other questions. If anybody has a last question, please put it in the chat or the Q&A. I'll ask one more. So what we're seeing too is it maybe not just public data, maybe you can comment on this, but there are companies, there are organizations that have gone after a target, gone after a therapeutic area, maybe failed in the clinic or whatever. There's opportunity to go back and take a lot of that data and reanalyze it, whether it's mode of action, major mistakes made in the analysis or whatever. Is that something that you guys do as well for clients or customers?
Are you talking specifically about taking their clinical data and reusing it?
Yeah, like not necessarily public data, but internal data, or a company wants to share that data, et cetera. Is that a use case that you guys are involved in?
Yeah, so it's a great question. We have focused mainly on enhancing the use of publicly available data, but you could, we do have a few use cases that we're piloting, applying those same ETL connectors to proprietary databases,
Okay.
so that we can help people do that. But conceptually, the problem is the same. It's just changing the input source and making sure that the models have what they need to be able to find that information.
I think that was my takeaway. One of my takeaways from today is it is the same, because we're seeing the same ratios of quality versus non-quality data before and after cleaning. So there's an expanded, I think there's an expanded use case here. It'd be really interesting to see how that evolves.
Yeah, if anyone's interested, send me an email.
Yeah. All right.
Oh,
well,
yeah, and Yogesh, didn't you have one for people who wanted to try out searching?
Yeah, I would just like to say that if any in the audience want to, give a shot to findability on Poly, if you want to try it by yourself for your biological question, then you can send me an email. I'm not able to send like message to everyone, John. I'll send it to you. You please send it there, and I'll be happy to get you started.
Okay.
Yeah.
Let's put that out to everyone.
Just that, so there is an additional L in the before the name.
Yep. Is that that's
Starts
the
from
correct
Y.
email? Okay.
Yeah, starts from Y.
Starts from Y. Okay.
Yeah.
Let's see if I can. There we go. I don't know how that happened. There's an extra L in my day. I don't know where it came from. Okay. All right, great. So we're at the top of the hour. Great job, everybody. It's exciting to see what you guys are doing. It's very needed, and as we have all know or are aware that the data environments need serious uplifting, and the fact that you guys are able to curate a lot of these public data sets, especially around an omics area, is really cool. So looking forward to all the future work from you. Great job.
Thank you.
Thanks so much, John. It was really fun.
Thank you.
All right. Take care, everybody. Have a great weekend.